the number of additional attendees is starting to slow down a bit. So yeah, we will begin this Q&A session. Thank you very much for tuning in. My name is David Montwe, and I'm an assistant professor in the Department of Forest Resources Management, the Faculty of Forestry at the University of British Columbia. And we just had 4 wonderful presentations by Claire, Mengjun, Susan, and Rafael, and I'm excited to moderate this Q&A. Please place your questions into the Zoom chat, but I will also do my best to monitor the session chat as well. So if there are any questions in there, we can also address them here. And I have some questions myself, but I would like to mainly give the floor to the audience. So thanks again to the speakers. And now let's begin with some questions. And the first question, it's from the session chat and was directed at Mengjun. The question is, what is the divergence between Loblolly pine and Ponderosa pine, since you are using Loblolly as a reference? There are some recent studies showing that variants in non-genic regions being associated with adaptive evolution, specifically in species with larger genomes. I would be curious to see how your results vary if you included the non-genic variants. And an additional part, this may specifically be important for the LFMM, for the estimation of the latent factor too.
Okay, great question. So for the first question, so for Loblolly pine, it's closely related to the Ponderosa pine. Both are classified in the subgenus the Pinus as opposed to the Strobus. And actually, before we ran calling the Snips, we tested multiple pipelines using both de novo and reference pipeline to compare the number of Snips they can get or the quality of the Snip they can get. And stacks with a reference genome as Loblolly pine can produce the largest number of Snips with the least proportion of paralogs. That's why we choose this reference genome and we use this pipeline to call the Snips. So for the non-genic ones, actually for the first try, when we first conduct the LFMM, we use all the Snips, the 4 million Snips to run this. And in this species, because they are like low disequilibrium, and yes, there are some papers showing genes in the non-genic regions can potentially affect the species performance, but we don't include that because in conifers, especially in Ponderosa pine, they have very low linkage equilibrium, and for the Snips that are located in the intergenic ones, basically there are false positives. That's why we filter that out before we ran that. So for the latent factor, actually we use all the 4 million Snips to get the latent factor with instead of using the filter one. So for the filter one, we only use that for the association analysis, not for the latent factor. I'm not sure whether that answered your question or not. Thanks.
Yeah, thank you very much, Mengjun. The next question is for Rafael. The question is by John Degner, and it was also asked in the session chat. Rafael, have you considered analyzing any other drought phenotypes within your data? For example, date at which FVFM drops below a certain threshold. While this would be categorical data rather than continuous, it might shine a light on a different component of drought tolerance than your slopes.
Yeah, sure. Thanks, John. Just before answering that question, I just would like to use this space very quickly to acknowledge Brendan Lind. I didn't have the time to do that in my presentation. A postdoc in our lab who did lots of great work and worked together with me to analyze the genomic data. So check out his GitHub page for all the pipelines and analysis. So John, yes, I also measured visual damage, and visual damage was highly correlated with the slopes that I observed that actually with the chlorophyll fluorescence measurements. I considered and did this approach of looking at the thresholds. However, when we look at the thresholds, one threshold that is critical for one provenance is not critical for another provenance. So I was not sure how to use that. And the other question about using sigmoidal curves, yes, after actually talking to you, discussing this with you a little bit, I did fit every individual with a beta regression, which kind of fits proportional data much better between 0 and 1. However, the slopes of the beta regressions were also highly correlated with my linear regressions, which are much simpler to explain and to explore. However, everything was super correlated. I also, after feeding the beta regression and looking at this sigmoidal curves, I looked at the inflection point, or the point in the curve where the decrease is steeper. And it's also correlated with the slopes there. But I feel with the inflection points, I definitely have more clarity in terms of comparing the provenances. So I hope this answers your question.
Thanks, Rafael. And the next question is for Suzanne from Esteban Galliano. Hi, Suzanne, great talk. Which program do you usually use for getting the gene family evolution and for the networks, HBEF integration, DNA damage?
Yeah, so for the gene families, all of my experience is pretty much using OrthoFinder and also Café on top of that to identify the rapidly evolving genes. So I know there are alternative pipelines to do that. I haven't had a chance to try those out yet myself, but I find that those tools work well in terms of giving me a good starting point to kind of delve into things deeper. So identifying like the larger trends and whatnot. And in terms of the networks, I use Cytoscape and somewhat StringDB, which actually is a really cool web application. I found very useful. I can pick like Arabidopsis as my baseline organism and just upload my sequences, and it does all of the pathways for me. It provides annotations, like functional annotations, and I can actually export that to Cytoscape. So that's been really helpful in sort of looking at those closely.
Thank you very much, Susan. The next question is for Mengjun by Marie Vance. Mengjun, any idea why you didn't find any overlapping Snips in your two analyses? I know there is a lot of missing data associated with the GBS approach, so I was curious if you were using the same set of Snips for both analyses.
Yes, so for the G2P, genotype to phenotype, we are also using the same SNP to run the association. We still don't know why we didn't find any overlaps between the G2E and G2B, but when we conduct annotation for the associated SNPs, their function overlap like a lot, including like ubiquitin or the absolutely exit signal way or the cell formation, which are directly associated with the drought tolerance of this species.
Thank you very much. The next question would be for Claire, and that's a question I'm curious about. At the end of your talk, you gave a little bit of an outlook about upcoming work related to wood anatomy. And I was wondering if you can expand a little bit about what features you plan to analyze and what results you might expect from that.
Oh, okay. That's a really good question. And basically, that's a lot of work. I started this project several months ago. What I can say is basically because I had the Silviscan data. So we had measurements of wood anatomy at really, really fine temporal scale. Okay. So I can look at the features within the growing season. And basically what I'm trying to detect is the peaks, it's a kind of frequency of peaks I have in my wood wood anatomy traits. So I'm just looking at wood density, like radial lumen diameter, other tracheate features, and I'm just developing at this stage methods to appropriate method to detect the different peaks within the growing season, and it's not easy at this stage. So I'm developing different methods in parallel, and I'm going to consolidate the results between the two methods I'm currently developing. But it's a lot of work, and I've realized that it's not easy, and maybe that's I'm kind of ambitious, but it's going to take some time to be able to detect these peaks properly, because there's no recognized method at this stage to detect peaks. We have a multiple packages in R, but it's I should say there is no conventional method and recognized method right now. So it's a lot of work.
Yeah, thanks for that outlook. That sounds like an very exciting part of the project. Then the next question is for Rafael by Jean Bousquet. Lots of hard work, congrats. You had very little overlap between significant SNPs from both varieties. From the genes involved in each group of significant SNPs, does it imply different physiological mechanisms, or is it that historical gene flow has been so low between the two varieties that SNPs are little shared between them?
Yeah, thanks, Jean Bousquet, for the question. So I think these are two great hypotheses to test, and maybe a combination of both. After testing them under drought conditions in the greenhouse, I noticed, the two varieties behave very differently under drought stress. And if we think about this hypothetical drought response, anisohydric or isohydric, I would definitely, categorize the coastal variety as a isohydric, where the plants for this variety control more or less the gas exchange in order to not to lose too much water under drought stress, and anisohydric for the interior variety, where plants, I would say, don't care too much about losing water because they have the morphological adaptations to deal with the drought. So two different strategies, it's definitely playing a role there because it's clear visually. But the lack of gene flow could also be a good explanation. And this we see with high levels of structuring or FSD between the two varieties. So, but I will definitely explore more on this, and I hope to come with some answers.
Yeah, thanks for that answer. A question for Suzanne by Natalie Isabel. How much time does it take you to conduct gene annotation?
I think so I work with much smaller genomes than many people here, so between 400 and 600 megabase pairs. And something like that, I would say like a month is a reasonable estimate, and that's sort of doing all of my normal pipeline, including repeat masking and preparing RNA-seq and protein evidence. And then I usually use a breaker, which will take several days, and some filtering after that. And using, if I have transcripts to compare to the gene models, and functional annotation, like including all of those steps. I'm pretty familiar with the process, so probably about a month.
Thank you. The next question is for Mengjun and Rafael by Funda Ogut. Great talks. Have you looked at the effect sizes of the causal Snips?
So I will start. So for my project, yes, because there's another project going on. We are trying to use the association Snips to as like a candidate genes to sequence them, and then we use other data to predict whether the seeds with this kind of Snips, this kind of genes, can perform better or worse in some specific environment. So we do have the data for the effect side, but I didn't show the results in this presentation. Yeah.
In my case, not yet, but we are going to investigate it further.
Thanks for that. So we're going, getting close to the end of the session. We couldn't answer all of your questions, but if you stick around, I would invite the speakers to type answers to all of your questions. Maybe as the last one, also for Rafael, by Antonio Castilla. Rafael, excellent presentation, seeing all the within provenance variation for drought hardiness. I just wonder how much micro-environmental variation exists in the natural populations. Do you have any insight on that?
Yeah, sure, especially for the coastal variety, considering that it's a more mountainous region with more slopes and more differences in elevation and precipitation. So lots of microenvironment things playing a role there. So this is one of the sources of the within provenance variation that I think is happening. So this would be one of the explanations. So the other one is staining genetic variation within provenances, lots of overlapping between provenances for the trait. And another one would be plasticity. But there is a very nice paper from the 80s, I think, in Coastal Douglas fir that analyzed that different features in topography and in a very particular region on the coast, and they saw a very high effect of those microenvironments or microtopography features on the adaptation to drought. So this is definitely playing a role, especially on the coast.
Thanks for that. That concludes our session. So thank you very much for attending, and looking forward to seeing you at the rest of the conference.
Thank you, David.
Thank you.